Next up, we have oral presentation 4, Galloway, New Jersey, Stockton University, Team 1.
Hello, everybody. My name is Valkyrie Falciani.
And I'm Hannah Sandler.
We are from Stockton University, and our experiment is called Spores in Space, the Effects of Microgravity on Endomycorrhizae. Our other principal investigator was Danielle Ertz, and our other investigators were Francisca Iqui, Ariel Pachel, Cindy Lowe, and Megan Pierce, who unfortunately were unable to attend today. Our faculty advisor was Dr. Luke. Mycorrhizae are a mutualistic relationship between fungi and plants. The fungi colonizes on the roots of the plants and helps the plant pull more nutrients out of the soil. This relationship is important on Earth and may prove to be useful for agriculture in space.
For our experiment, we used the Type 3 FME Mini Lab. In Chamber 1, there were 6 flax seeds. The mycorrhizal spores were located in Chamber 2, and there was water to activate the experiment in Chamber 3. This experiment was flown aboard the International Space Station in August of 2017, and 2 identical experiments were conducted on the ground around this time.
Once the experiment returned, it was opened, and no plant growth had occurred. The inoculum containing the spores was separated out and combined with fresh seeds. Other post-flight experiments were also conducted to isolate the variables. These other samples included fresh seeds and fresh spores to be used for comparison. All of the samples, including the ground truth samples, were fixed using a standard plant fixative and stained using Chlorazol Black E. The samples were analyzed using the Lenka DM750 microscope and photographed using the attached Lenka ICC50 HD camera.
We found that the spores exposed to microgravity conditions have produced condensed and localized growth patterns, as well as reproductive structures than the ground truth experiments. An example of a reproductive structure is shown in the top right photograph. Our main goal while looking under the microscope was to see if any infection from the spores was present among the seeds, and if there was any difference among all parts of the experiment while having the infection present or not present.
In conclusion, space travel does affect the growth of arbuscular mycorrhizal fungi. We believe this is caused by the stressful conditions the experiment underwent from the journey up to the International Space Station, exposure to radiation, and the microgravity conditions aboard the International Space Station. However, more work needs to be done to determine which of these factors is the main stressor, and if this is a viable option for agriculture during prolonged periods in space.
We would like to thank Stockton University, the School of Natural Science and Mathematics, the School of Education, the School of General Studies, and the Stockton STEM Collaborative for this opportunity. And a special thank you to Dean Straub and Dr. Luke for all their help and guidance. Thank you. Do you have any questions?
Okay, no questions. Thank you,
All right,
ladies.
thank you.
Thank you.